just as a brief introduction, this is the Spanish Researchers in UK Association. We do this kind of events where we want to make science closer to everyone. And Jenny has very kindly accepted our invitation. Her presentation looks very, very promising. And just as a very brief description, Jennifer Batson is the principal scientist at Exonate, which is a biopharmaceutical company working on drug discovery for retinopathies. So Cristina here is going to talk a little bit more about this. But just to let everyone know, we are recording this and we will stop recording just before the presentations at the end, the questions at the end, sorry. So yes, just for all of you to know. So well, thank you very much. Cristina, over to you.
Hi, everybody. So thank you again, everybody, for joining this session, and especially thank you, Jenny, for doing this. I think it's a real pleasure for me to introduce Jenny to the second part of Women in Leadership, because I had the pleasure to work with her during the last 4 years in Exonate. And I think it's a clever scientist, is very determined, and I did learn lots from her, especially, well, many things. She's very insightful, but especially, I think I learned whenever you fail in an experiment or something goes wrong, she always let you focus in whatever you can get good for that, and what can you learn, and extract the best of whatever was done, and always learn something from that fail and try to improve for the next one. So it has been a good experience in all senses. So just very brief introduction, because I think she is going to tell you more about it. She did a bachelor's degree in biology in Manchester, with 1 year experience in industrial position in, I'm not going to attempt to say the name, in Vienna, in Boehringer Ingelheim, or something like that.
Pretty good.
Yeah. And then she did a PhD in the University of Bristol. After that, she moved to Nottingham for a postdoc with Professor David Bates. And after 1 year and a half, she was key in setting up and developing Exonate, the company. She is working as principal scientist now. So that's it. Thank you very much, Jenny, and all to you now.
Thank you very much, Chris, and thank you for that lovely introduction. Do you need to enable me to share my screen?
Carlos?
Oh, it's working now.
Okay.
Okay, yeah, so thank you very much. It was really useful for me actually to have time to reflect on where I've got to so far. So I'm principal scientist at Exonate, as Chris said, and Exonate is a small biopharmaceutical company based in Nottingham and Cambridge in the UK. And so I'm going to talk to you about what I've been doing in leading the biology team. And we've been focusing really on developing eye drops to treat diseases of the back of the eye, which are retinopathies, and those are the leading causes of blindness in aging and in the developed world. So I should say no one has been able to make an eye drop, and pretty much everyone thinks it's impossible to make an eye drop for these diseases until now, hopefully. So Exonate started as a spin-out company from the University of Nottingham in 2013, 2014. And in the 7 years since then, we set up the company, started the drug discovery program, and we've taken the lead drug into the clinic. And we've been able to do that through a collaboration recently with Janssen Pharmaceuticals. Janssen are the pharmaceutical part of Johnson & Johnson, who are one of the biggest pharma companies in the world. So that's a real feat that we've been able to collaborate with them, and we're really enjoying working with them still. So as a company, we have 16 employees and 9 are female, including our CEO, Catherine Beach. So we're proud that we're bucking the trend for female founders, investment and team equality. And I do believe that diversity and gender equality are really important for a strong team. So especially in drug discovery, we have a lot of problem solving to do. And I think that the best way to approach problem solving is using creativity and using different inputs and diverse ideas. And also for innovation and to continue to develop, we need to be challenging the current thinking. So we need different ways of looking at problems. So this evening, hopefully I can talk about my career journey and with some insights into leadership and drug discovery along the way. So as Chris said, I started out at University of Manchester with a degree in biology with industrial experience. And I really chose that because I actually wanted to work abroad as part of my degree. And this was the best way that I could see to do that. So I chose biology because it was a very broad base and it meant that I could pick and choose what subjects I wanted. So at one point I nearly switched to pharmacology, which would be the study of drugs and how they behave in the body. And at that point there was a collaboration with Boehringer Ingelheim, who are a German company based in Ingelheim in Germany, but they also have a smaller group in Vienna in Austria, who are a cancer research centre. And that was really set up for the pharmacology students. So I had to sort of push a little bit to be able to go on to that, but they let me go and that was absolutely great. So at Boehringer Ingelheim, that was my first real lab experience. And I was involved in two different projects. One of them was compound screening at quite an early stage. And the other one was a lot later, looking at tumor inhibition with a compound that was then called the IBF 1120. But it's now marketed as nintedanib or OLAF or Vargatef, and it's approved in non-small cell lung cancer and in idiopathic pulmonary fibrosis. So I put that there not because I think I really contributed to that drug development, but because I think it shows that I was kind of lucky to be in a drug development team that was being successful, and it allowed me to really learn good techniques and lab skills right from the beginning. And what that meant was when I came back to my final year project in my degree, I could really hit the ground running. I put this picture here because it amazes me how much labs all look the same. So this is a biology lab in Manchester. It looks exactly the same in Boehringer. And I've also been to labs in the States and in Singapore, and all the labs look the same, the dynamics are the same, and it's still the same arguments over what's on the radio. After Manchester, I moved to Bristol and I did my PhD with Professor Kate Nobes, who's here. And this was a Wellcome Trust funded PhD, which is relevant because the first year consisted of 3 different lab rotations. And I was really lucky there because they're only 3 or 4 months each. And I managed to publish 2 papers out of them. The first one was this one in Kate's lab, which was really because of a postdoc, Jono here, who at the time was finishing up and was just finishing this paper, which was quite a lot of work. And it was based on a really old cell biology observation that when cancer cells collide with one another, they repel and move away. But when they collide with normal cells, they kind of invade, and that was thought to contribute to cancer invasion. And that was observations that were based on work in 1958. And Jono had looked at that and figured out the mechanisms that were involved and that it was driven by EPH receptor signaling. So I came in and did one experiment that showed that CDC42 was involved downstream of the receptors, and that got me onto this paper, which by the time I'd come back to actually start my PhD properly was published. So that was a good start I had to live up to. So I wanted to look at the mechanisms involved in cell migration and also what that meant for cancer invasion or dissemination. So this is mostly what it consisted of, was a lot of imaging and just looking at the cells as they migrated together. These are just static images over time. But these are 2 prostate cancer cells, and you can see that when they interact, they move around and repel from one another and move away. Whereas I found that if you knock down signaling proteins such as VAV2 with siRNA techniques, when the cells interacted, they then didn't repel, but they just kind of merged past one another and invaded their space. And that had implications because when you knock down EZH2 or the EPH receptors that reduced the dissemination away from a tumor spheroid. I think at this point, because Jono had left, I was able to work quite independently in the lab. It was only really me in the lab. And that meant that I could set up new techniques. So this was based on a conversation I'd had at a conference. And it was a new way of injecting cancer cells into a 3D gel to look at the spheroid growth. So after my PhD, I moved to Nottingham to join Professor David Bates, who had just moved his lab there from Bristol. And in Bristol, he joined with Athena, doing a PhD there, that a protein called SRPK1 was involved in prostate cancer cell growth. So this experiment here shows tumor growth in controls or with an SRPK1 knockdown that depletes the protein. And you can see that the SRPK1 knockdown reduces the tumor growth. So I was really interested in this, and it was known that SRPK1 is upregulated in numerous cancers, and that it regulates a process called alternative splicing, which is where different isoforms are produced from a pre-mRNA template. And that creates diversity and can be dysregulated in cancer and forms different isoforms that have been shown to contribute to the different hallmarks of cancer progression. But the mechanisms and the splicing events that were involved were still unclear. So what I wanted to do was to figure out what SRPK1 was doing, what splicing was changed, and how that led to this difference in tumor growth. So I was invited to write a comment on this paper, which I did looking at how the SRPK1 inhibition changed the splicing of a couple of known targets that were published in the paper. But I wanted to write fellowship applications, and I was really interested, based on my PhD work, to look at prostate cancer invasion more. And I wanted to look at lymphatic invasion. This was all based on a conversation with Professor Helga Vig at a conference in Bristol, who had expertise in lymphatics and access to tissue. But also I wanted to go to California, so I recruited Yang Fu onto my fellowship applications, who is a real expert in splicing, and showed that some of the really early splicing factors were involved in changing the different isoforms. So I was surprised actually when I look back at this, but I submitted 4 different fellowship applications, and these were quite big fellowships. The Henry Dale Wellcome Fellowship went out for review, but was considered overambitious. And none of these actually got funded. Looking back at it now, I can see why. But I did learn useful skills during this in setting up collaborations, grant writing, and project design and costing. I think what I hadn't learned was how to set up a project that was actually going to deliver, and how to get a successful project. So while I was writing fellowships, I was actually supposed to be working on a postdoctoral fellowship, which I was doing and spending most of my time on. But this was a Hermes Fellowship for Outreach, Innovation and Business Engagement for 9 months. And the aim of the fellowship was to secure a business plan for Exonate and to raise investor confidence so that we could start the company. So I was responsible for setting up experimental procedures in the lab so that we could test new inhibitors. And I also worked with the CEO to commission market research and to engage with key opinion leaders who are experts in the field and investors to develop this plan. And also identify questions that were causing doubt for investors and were unanswered, and potential for differentiation. And essentially, this was filling the void of those skills that I didn't have in the fellowship applications. I had to learn them now because we needed to put them into practice for a business plan. And that meant that we had to put the project together, outline how it was all going to work into a level that would give investors enough confidence to invest. So starting a company is quite a risky process, and there were several risks involved in it. For me, 9 months for a first postdoc position isn't very long. So the ways I was kind of mitigating that risk was also applying for fellowships at the same time, and applying for investor funding, but I don't think I thought that it was ever going to be 9 months. And that's because the confidence I had in potential for extending it, which was based in how I thought the project could progress with Dave and Catherine, the CEO. And also because Dave was setting up a new lab, I thought there would be potential for expansion there. I obviously had no experience of setting up a company or pulling together a business plan, but that was made possible by working with Catherine, our CEO, who does have fantastic experience in this. And I was able to build on previous experiences because I had set up new lab techniques during my PhD, so I could apply that to new lab techniques for this project. But despite all of that, startup companies do usually fail. I found a paper last year where it was published that it was only 23% success rate. So generally, the likelihood is that we would fail. The mitigation for that is if you're going to fail fast. So Catherine's approach is to do the killer experiments, and that means you want to kill the project if it's going to fail, or kill the parts that aren't working, because that means that you can find the best route forwards and focus on the experiments that are going to work. So I think that probably comes back slightly to what Chris was saying on finding something to learn from a failed experiment, is that a failed experiment can always tell you something, even if it's just don't do that again. So the other thing that I thought was useful to consider here is actually what the real risk benefit is, because there should always be a benefit if there's a risk, and well mitigated risks should come with rewards. So yes, there's a big risk. Usually startup companies fail, but if we succeeded, then that would be really successful and could lead to really exciting things. Also, on a personal level, the risk that I wouldn't learn anything or wouldn't develop any skills during this process was really low. So for me, probably I was more interested in learning new skills and developing, so I probably wasn't going to risk not getting that. But putting this together, I think I realised that I might be a bit of a risk taker. So in 2017, I joined a sailing race across the South Atlantic Ocean. This is the Clipper race. It goes all the way around the world in different stages, and I joined the South Atlantic stage. This is quite dangerous. These are 70-foot yachts. And this looks like we're having really nice sailing here, but actually I think what we're doing is evaluating some damage that's occurred up here. I think this photo was taken right before this photo, where you can see there's a lot of sail wrapped around here where it shouldn't be. And what happened was we were flying a sail that was absolutely massive at the front of the boat, and it had wrapped around this wire that goes all the way to the top of the mast and had started to unravel it. So that was a bit of a problem. What happened after that was we tried to cut it down, and all of the sail went in the water and then started going under the boat, which is also not ideal. So this photo was taken just when we'd managed to pull that sail back up onto the boat, and we all had massive smiles on our faces because we'd managed to fix the problem. This photo is our skipper Bob and me and Nathan fixing the inner forestay, which is this wire that goes all the way up there. We actually had to unravel it because it was all twisted up and put it back together. So high risks here. There's always mitigation processes. You can see that we're all wearing harnesses and life jackets and we're clipped on. But probably the way that we overcame this problem was because Bob has all the experience and knows how to fix broken boats and make everyone smile when it's an all hands on deck situation. So the learning points there were that always have a mitigation plan and know the risks, and then you'll be able to work your way out of them. But also I think you can learn useful and interesting skills outside of work as well as inside that can apply at work. So after this, in the last couple of years, I've trained to become a sailing instructor, definitely not in this size boat, but in smaller dinghies. And whilst doing that, I've learned the difference between instructing and coaching. And coaching skills that they teach us to use are about trying to identify and develop a growth mindset. And what that means is that you develop a mindset where you look at challenges as opportunities. Failure is an opportunity, and it's about looking at where you can make things work. If you don't know how to do something, like I didn't know how to start a company or develop the techniques, you can just break it down and approach it in a way that you will learn it, and then the reward from that is much greater than if you hadn't have worked it out. So in drug discovery, there are also big risks and mitigations. So this is from a study in 2014, which I would recommend everyone to read if you're interested. It's from AstraZeneca, and they reviewed all their small molecule drug projects from 2005 to 2010. And they found that 60 to 70% of preclinical projects are successful, but something terrible happens in phase 2, and this drops down to 15% for AstraZeneca and 29% for the industry median. So they looked at this and broke it down, and they found that the main reason for failure in the preclinical stage is a safety problem. But as that progresses, the reason for failure is more due to efficacy, which suggests that they haven't selected the right targets for their drug development program from the beginning. So to address this, they established the 5 R's framework, which is the right target, right patient, right tissue, right safety, and right commercial potential. So what we were doing when we were setting up our business plan was working out how to bring all of these different aspects together to be able to start the company and to generate confidence that we had all of these in place. So what we were actually going to do. These diagrams show VEGF pre-mRNA, and Dave, while he was in Bristol, had discovered that there is a different splice isoform of VEGF, which is generated for splicing into the distal exon called 165B. So these isoforms are quite similar in structure, but they have opposing functions. So VEGF-A165A is the traditional angiogenic isoform that causes blood vessel growth and leakage. But the 165B isoform is the complete opposite and opposes that and has anti-angiogenic and anti-leakage properties. It also has additional beneficial properties. So these are both forms of the same VEGF gene. They're both endogenously produced, but in disease situations, VEGF-A165A can be upregulated, and a switch in this balance can cause the angiogenesis to occur. So David shown that this splicing process is mediated by this kinase called SRPK1, which phosphorylates a splicing factor SRSF1, and that produces the pro-angiogenic disease-causing VEGF. But if you inhibit SRPK1, then that balance switches, the splicing is different, and you get the anti-angiogenic isoform. So that's all very well, but VEGF is involved in quite a few different diseases, such as cancer. I already showed you data where SRPK1 mediates cancer growth, kidney disease, eye disease, pain. And so which disease should we focus on? So previous work in Dave's lab by Mel had looked at SRPK1 inhibitors in an angiogenic model of wet age-related macular degeneration, which is a retinal neovascularization model. So she had used SRK-340 and Sphinx, which are 2 existing early stage inhibitors, but they're not really suitable for drug candidates. And what you can see here is with topical eye drop administration, there's a reduction in the leakage blood vessel growth with these eye drop administrations. So this is really important because no one had been able to make an eye drop to treat these diseases. So could we develop a drug discovery program from these starting points and could we use retinal neovascularization as our disease target? Well, retinal vascular diseases are the leading cause of blindness, and you can track that by non-invasive imaging of the human eye. So this is a normal person's eye. You can see blood vessels, and this is the macula, which is responsible for central vision. But in a patient with wet age-related macular degeneration or with proliferative diabetic retinopathy, with fundus imaging or with fluorescein to look at leakage, you can see that there's new blood vessel growth and leakage into the eye. And these diseases are driven by inflammation and hypoxia or low oxygen levels, which cause release of the pro-angiogenic isoforms of VEGF. So in wet AMD, that causes choroidal vascularization through the Bruch's membrane and into the subretinal space. In diabetic retinopathy, you get leakage and blood vessel growth in the retina layers. But both of these lead to disorganization of the photoreceptors and leakage of fluid into the subretinal space. And that causes central vision loss. And in wet AMD, if that's untreated, then patients will lose their vision within 24 months. So the good news is that these are treatable. So this is a really bad example of a lot of leakage on a cross section of the eye, of the retina at the back of the eye. And this is the same after treatment with an anti-VEGF injection. So there are anti-VEGF antibodies such as Lucentis or Eylea, which have been shown in clinical trials to stabilize vision in 95% of patients and to actually improve vision in 40% of patients. So it looked like retinal vascular diseases could be a good target. You can follow the disease process. Anti-VEGFs work, so VEGF is a validated target, but there's still big disease markets. 35% of patients with diabetes will go on to develop proliferative diabetic retinopathy, so that is a big market. So we thought that we would focus on retinal vascular diseases for our drug development program. Additionally, back to the mitigation idea, this does de-risk things slightly because it's a much shorter clinical development plan. So you should be able to see efficacy at the back of the eye within months rather than years for some cancer programs. And you can image that. So what's wrong with anti-VEGFs then, if they do actually help with the disease? This is a graph which shows two different clinical trials, and it shows you the letter score, so an indication of the vision quality during the clinical trial for the initial study with aflibercept or ranibizumab. And you can see that the vision improves during the clinical study, but then afterwards, in 2 years follow-up after the clinical study ended, the vision returns to baseline. And this is despite these patients still having treatment. And then by 7.3 years, the vision has actually worsened. So the reasoning for this is not entirely clear, but it's thought to be partly due to compliance, because these anti-VEGFs have to be given by intravitreal injection, which is directly into the eye, and it should be once a month. So it would have been once a month during the clinical trial. But that's an invasive process. Patients have to go into clinic, and it's also really expensive. So the general practice is to increase the treatment period to extend it. So the treatment schedule would have been lower during the follow-up period. And a third of these patients also went on to develop macular atrophy and fibrosis, which are bad outcomes. So if we could develop an eye drop that would change VEGF but not completely inhibit it and restore the endogenous balance, then that would be address a serious unmet medical need. And some of the clinicians we actually spoke to said that it would be a holy grail in this field. So we thought that would be a good thing to do. So our goals were to introduce a revolutionary game-changing eye drop for the treatment of these diseases, to use in-house expertise to deliver effective doses to the retina, and to generate a more effective therapy with a new way of changing VEGF. So this is a bit of a challenge. I haven't shown you that we have any expertise in delivering effective doses to the retina yet. And this was a big challenge. So how could we succeed in making an eye drop when all the big pharma companies have already failed? And examples of this are GSK with the pazopanib, and since then, people keep failing, Novartis and Bayer. And really, the problem with these drugs is that they're all repurposed anti-cancer drugs. So the question is, how can we not take something that already exists, but actually design something so that it gets to the back of the eye, so that it can have efficacy? And the way that we went about that was by talking to experts, clinicians and experts in ophthalmology, find out why those drugs failed and approach the problem from a different angle. So we wanted to start from the beginning and work out how to get a drug into the eye. Delivery to the back of the eye is challenging. So this is the front of the eye here, and this is the lens, which takes up a big amount of space. And this is the back of the eye where we need our drug to get. So you have to either go through the cornea and then get around the lens, or you have to get your drug around the back of the eye and then through the transferal route. And no one knew how to do that. So the way we set it up was to obtain eyes from a local abattoir that weren't being used, and clamp them in these permeability chambers. So this is what one of these eyes looks like. We dissect the eye, because I hadn't done this before. It took us a while to figure out how to dissect the eye without losing the retina, which is this slightly opaque pinkish region there. But we've got the full thickness, clamped it in the scaffold, and then we could look at how the SRK1 inhibitors were getting through the different tissue layers. So our goals and what that allowed us to do were to develop inhibitors that had high permeability to get into the eye, so they could have exposure and duration wherever they were needed. But also at the same time develop the potency so that they were really active against SRPK1, and so that they could switch that VEGF balance back to the endogenous anti-angiogenic forms. We would need to generate safe drugs. I showed you that that was a big reason for failure in preclinical studies. This is less of a risk if you're making an eye drop, because really the systemic levels in the blood and systemic organs would be very low, but we would still need a highly selective non-toxic compound. And since VEGF is a validated target, that is a real strength, because it shows that we would be on the right track and there's precedent. But it does show that we would need differentiation. So mainly that is in an eye drop versus an injection, but we need to bring that into our presentation. So I hope you can kind of see this. I just took it from the ABPI. It outlines the different phases of drug discovery, but what I thought was really interesting also has the number of candidates at each stage, the cost, and the timeline. So in drug discovery, apparently, usually you would have 5,000 to 10,000 drug candidates, would cost you 436 million and take 4 to 5 years. We obviously did not spend that much because we wouldn't be able to raise that much money. But we did that because we didn't want to look at that many candidates. So we took a different approach to some programs by crystallizing the structure of our protein. And that meant that we could rationally design compounds, and we had quite a fast design make test cycle. So we would make a set of not that many compounds, and then we would test them on our biology. And then we would feed that back to the chemist and say, well, that worked, that didn't work. Make us a few more, please, that have those benefits. And we did that, and that's how we got a lot further with a lot less compounds. But it was clear that we would still need a lot of money and a lot of investment to be able to get into the clinic. So what we needed to do was to establish a target candidate profile, and that is a document that outlines all the key drug properties that we would need to hit. So the permeability, the potency that I showed you, and then also the next stage on from that, which is the target drug profile, and that defines the end product which would go into the clinic, the intended disease and the intended patients. And it's good to start that early and to begin discussions with regulatory authorities early, because that defines the data that you need to put into regulatory submissions. An interesting thing I discovered while putting this together was that aflibercept, or Eylea, which is one of the anti-VEGFs that is approved for these diseases, actually was one of the worst on this scale. So usually it takes 12.5 years to develop a drug and costs £1.15 billion. Eylea took £3 billion and just as long. Actually, is probably worth it for Regeneron because it was the 5th biggest sales drug last year. It brought in $8 billion in sales, and it makes up 70% of Regeneron's sales. So they are really reliant on it. And when it goes off patent, they're going to be slightly scrambling around for something else. So the way that we were able to raise enough money to get through that preclinical phase was in that we got a Wellcome Trust Seed and Drug Discovery grant. This was all the way from late optimization, where so this was our first milestone, where we would need to find 5 compounds that met that target candidate profile that we'd set out. We would then need to nominate one of those compounds and work out how to formulate it, and that gives us our preclinical candidate. And even after that stage, there's still quite a lot of work to do to really demonstrate that that candidate is going to be safe. Has to go through very strict good laboratory practice studies in order to get enough data to put together a submission for clinical trials. So this Wellcome grant was absolutely amazing because it allowed us to take our compound all the way through preclinical development, ready for the clinic. So keeping in tradition with all our other grants, this wasn't successful on the first application, but they asked for us to work on it further and then resubmit. And then we were awarded £4.9 million for 3 years. And I can remember when Dave rang me to tell me that we were successful. I nearly fell off my chair. But this enabled us to grow, and we were able to build a team from 3 scientists at that point to 6 and develop the compound. We recruited a great team of scientists to do that, and each bring unique skills that they can contribute to the drug discovery process. And I think that we've worked hard together to create a good team environment. So none of this wouldn't be possible without that good team environment and the good team. And it's really critical to the success. I think everyone also has leadership skills and can show leadership skills within the team and in different roles and in and out of work. Because if you think about it, leadership is about inspiring and helping others, and we all do that. So another part of enabling a good team is giving them the tools to be able to work with, and for that, data management is a key aspect. So luckily we worked out early on that we would really need to protect our intellectual property if we were to be able to sell it later on or develop from it, and that would be critical to investors in future partnerships. So we worked with Dotmatics, which is an online cloud-based software system which can register all of our compounds and keep track of all our drugs and all the data associated with them. But we had to set it up. So I had to work with the software engineers at the beginning to set up processing scripts and data tables that would store all that data. and the electronic notebooks that we would use for record keeping. And I've just put this on here because it is a bit of a challenge, and it's something that I've just overcome a big obstacle this week. So it required a lot of understanding how the software is set up and how they want you to write the different programming to make it work. But I think we're getting that now, so that's been a good learning curve. In terms of conferences, at the beginning, we would go to conferences such as EVER, which is often in Nice, ARVE, which is the big American eye research conference. And we would present data, and we would be there really to learn about new science and techniques and see what everyone else was doing. But from the beginning almost, we met with pharma companies at the same time, which is an interesting way of going to conferences. There's a whole other world that happens at conferences where all the pharma companies meet. And we would pitch our ideas to them, and they usually sat there and humoured us. But in the last couple of years, they really started getting interested, and we initiated talks that led to our collaboration with Janssen. So that was probably quite a long thought out process, which really just worked out in the last year. So also, while we were at our conferences, we met with our clinical advisory board, and that was really important because they're the experts that are guiding our knowledge in the retinopathy field and how we were going to develop our clinical trial. Also, the British Pharmacological Society we go to every year, and I put that on because I was lucky enough to step in for Catherine at one point because she was at a different conference and join a panel for hot trends in drug discovery, which was a great experience for giving a presentation and sitting on a panel board. And similarly, I also was lucky enough to go to Bermuda because Dave couldn't go at one point. So I was always kind of ready to step in if someone couldn't go to a conference to go and give the presentation for them, which worked out well. So after all those conversations with the pharma companies, which we always learned something from, and with our clinical advisory board, the package that we ended up with, we pitched as a novel approach to revolutionize treatment for retinal vascular disease. And it was all based on that ex vivo transscleral modeling, where we designed inhibitors that we thought would get to the back of the eye. They had high potency and permeability, and we generated the confidence that they should work as an eye drop. And we backed that up with lots of preclinical studies and showed that it did correlate with levels in the back of a large eye. The differentiators that we pitched for our technology were that we could deliver those eye drops, they would be efficacious. It was a unique approach to designing drugs involving different medicinal chemistry, the ex vivo model, and that was verified by the scientific studies that we had done. And that this is a different disease approach, a different way of making drugs because it's mRNA targeted. It targets that splicing mechanism by inhibiting SRPK1, but then by switching the isoforms so that you get that healthy anti-angiogenic 165B, which would inhibit the diseased neovascularization. And then, of course, there would be potential for line extensions with different formulations, but also if you're developing an eye drop rather than an invasive injection, then there's potential for delivery to the contralateral eye. So it is known, in fact, in that clinical trial I showed you, that I think a third of patients had the disease phenotype in the other eye that they hadn't had at the beginning, but it often develops in the other eye as well. And we had strong intellectual property around our science, which we have protected. So that really led to collaboration with Janssen. This is a tweet that I took, which is pretty cool because they said that they're strengthening their leadership in retinal disorders by collaborating with Exonate. Awesome. And this is just a snapshot of our clinical trial, which is in process now. We are treating patients with our lead drug, so it's really exciting. This is mainly a safety trial, as phase 1s usually are, but we may also get some indication of efficacy, so we're all watching that space. And then finally, out of all of that, at the same time, I was awarded this BPS Outstanding Young Investigator Award, which was amazing. But I think what I would say from this is, if you're a member of a society, have a look and see if they've got any nice awards you can apply for. I've been a member of the British Pharmacological Society for years, and I just spotted this and applied for it years ago. And then one day out of the blue, they emailed me to say that they would like to give it to me. So this is great, because they gave me £5,000 just so that I could go to Sydney in Australia to attend a couple of conferences. But because our clinical trial is in Australia, also meant that I could meet with investors and clinicians just before we started the trial. And it also let me go to different labs just to visit them out of interest. So I went to see Professor Vicky Avery and Professor Ron Quinn at the Griffith Institute for Drug Discovery, just because I was interested in what they did. But I wanted to see their high content imaging. They have fragment drug discovery, and also the largest collection of natural products in their nature bank that they use as starting points for drug discovery. So that was really cool. And then the Australasian Society of Pharmacology and Toxicology in Queenstown, New Zealand. So then I'd just like to acknowledge everyone at Exonate, obviously the Wellcome Trust for funding, and Janssen. This is us on a team building sailing day, and this is us in the lab. And then thank you for listening.